Hi, this is Dr. Vijay. Today we are going to study isoenzymes. This is just an introduction about isoenzymes. Isoenzymes are also known as isozymes. These isoenzymes are multiple forms or isomers of same enzyme. That means these isoenzymes catalyze same biochemical reactions. For example, here a substrate A converted to a product B catalyzed by enzyme 1, and same biochemical reaction can be catalyzed by enzyme 2 also, or in some cases enzyme 3 also, or there may be an enzyme 4. All these enzymes are called enzyme 1, enzyme 2, enzyme 3 and enzyme 4. They are called isoenzymes or isozymes because they catalyze same reaction where substrate A converted to product B. The same enzyme has got different isomers. Usually these isoenzymes contains more than one polypeptide chains, or we can call it as subunits. Because of these subunits, they exhibit isoenzyme properties. Usually they are called heteromers. And these isoenzymes can be encoded by single gene or different genes. So these isoenzymes might have their gene in different loci of different chromosome or same chromosome. And also these isoenzymes can be expressed by different tissues, like in case of amylase, amylase can be expressed from the salivary glands as well as from the pancreas. All enzymes being a protein, especially in case of isoenzymes, even though they catalyze same biochemical reactions, but they differ with respect to the primary structure, that is composition of amino acid. So different isoenzymes of same enzyme activity might contain different number of amino acids, that is the amino acid composition. Even their amino acid sequence might be different. They might have different amino acid sequence, yet they catalyze same biochemical reaction. Since their amino acids sequence are different, because we know that there are different types of amino acids, they contain different R groups, maybe acidic, basic, polar, non-polar. So their electrophoretic mobility are different. So we can use this property to separate isoenzymes from the serum or plasma. And because of their amino acid composition is different or amino acid sequence is different, their Km value is different. The specificity for substrate are different for different isoenzymes of same enzyme. They might have different Km, different Vmax. In other words, the specificity or affinity for the substrate might be different for different isoenzymes of same enzyme. And because their amino acid composition and sequence are different, their heat stability are different. That means the ability to denature these enzymes may be at different temperature level. So again we can use this property to some of the isoenzymes, especially alkaline phosphatase. And not only kinetic properties are different, these enzymes has got their own inhibitors, or they can be inhibited by different inhibitors. Maybe one isoenzyme of the enzyme can be inhibited by one inhibitor, but this inhibitor cannot inhibit other isoforms or isomers of same enzymes. And they have different cofactors, like they might require different coenzymes or metallozymes. We will see some of the example for isoenzymes. First and foremost, creatine kinase, a very important enzyme. It has got 3 isoenzymes. Abbreviation for creatine kinase is CK. Previously it is also known as creatine phosphokinase or CPK. Nowadays we use CK as abbreviation for creatine kinase. It has got 3 isoenzymes, CK1, CK2, CK3. I will make separate videos in future for these different isoenzymes. Another example is lactate dehydrogenase. It has got 4 isoenzymes. The abbreviation for lactate dehydrogenase is LDH. LDH1, LDH2, LDH3 and LDH4. So in the future I will explain about these isoenzymes in separate videos. Other example is alkaline phosphatase. It has got many isoenzymes because it is expressed in different tissues. Bone expresses alkaline phosphatase. We call it as bone alkaline phosphatase. Abbreviation for alkaline phosphatase is ALP. Placental alkaline phosphatase, liver alkaline phosphatase, intestinal alkaline phosphatase, and even in the kidney there is an isoenzymes of alkaline phosphatase. So there are different isoenzymes of alkaline phosphatase. By using kinetic properties, that is different Km, heat stability and electrophoretic mobility, we can separate these enzymes. Amylase also has got 2 isoenzymes, that is expressed from the salivary glands, salivary amylase, and even we have amylase released from the pancreas, that is pancreatic amylase. So isoenzymes or isozymes are multiple forms of same enzyme and they catalyze same biochemical reaction, even though they are released from the different tissues or organ, and they differ with respect to amino acid composition, amino acid sequence, electrophoretic mobility and enzyme kinetics, that is they have different Km value. Thanks for watching.